YouTube. Welcome, Alina.
Thank you very much, Amina, for the introduction, and thank you everyone for attending my talk today. So as Aminath mentioned, my presentation today is about interpretable machine learning for chest X-ray diagnosis and COVID-19 detection. Before beginning, I'd like to acknowledge some of my collaborators in this work. So Harrison Layton, Danny Wang, Richard Wainwright, and Dr. Sally Shrapnel. And I'd also like to acknowledge some of the funding support I've received from the UQ AI Collaboratory. So to begin, I'd like to acknowledge the traditional owners and the custodianship of the lands on which we meet today. Here where I'm presenting from in Brisbane, that is the Yuggera and Turrbal people. I pay my respects to their ancestors and their descendants who continue cultural and spiritual connections to country and recognise their valuable contributions to Australian and global society. So I guess I've already received a little bit of an introduction about myself, but I thought I'd just give a couple of examples of some of my research interests, a little bit about what I do. So I'm a computer vision and machine learning researcher at the University of Queensland, and specifically within the School of IT and Electrical Engineering. And a big part of what I'm interested in is data visualization and understanding and interpreting neural networks. Part of the goal of that is to be able to develop robust and trustworthy AI systems. And I've had quite a diverse career to this point so far. So as you would have heard Aminath mention, I've worked on different things, for example, sports analytics, also looking at attention in driving, and more recently in medical imaging, including a new modality of electromagnetic imaging. And in terms of this talk today, some of the work I've done in chest X-ray analysis. But putting all these things together, what I really like to do is get an understanding of how machine learning models work. So as an example, on the right here in this animation, together with some of my collaborators, we developed a model that is able to predict the perceptual load or the demands of driving for a human driver. So here you can see that it's increasing when there's, I guess, more information happening or there's vehicles in close proximity. But just having this measure which says, more or less is not very informative. What we really want is to have more information behind how models come up with these kinds of things. And so now moving towards medical imaging, we've had a lot of success of AI methods, and they offer a lot of great promise for improving the speed as well as the accuracy of detection of diseases from images. On the right of the slide, you can see a couple of examples of where artificial intelligence methods like computer vision and machine learning have been applied in medical imaging. For example, detecting masses in mammograms, detecting lesions in the brain, looking at leak detection in the airways of the lungs, looking at the retina and detecting any kind of abnormalities, and so on. So within the space of medical imaging, COVID-19 is another type of application of where we can apply these techniques. And with the onset of the global pandemic, a lot of research fields pivoted their research towards finding solutions to the disease. So machine learning and computer vision was no different, and many researchers pivoted towards applying techniques towards diagnosing COVID-19 from chest X-rays. In fact, within the first year, there was already 320 papers about detecting COVID pneumonia from chest X-rays. So that's up until the point of the 3rd of October, 2020. Now we're even more than a year beyond that. So the amount of papers now is probably exploded exponentially in this space. So what my talk is about today is looking at some of the flaws behind COVID detection from chest X-rays, and specifically how we can use interpretability techniques for investigating that and understanding our neural network models. And so to begin with, I want to give a little bit of motivation for why you would even use chest X-rays for COVID diagnosis. So as we probably all know today, typically we diagnose or detect COVID using PCR tests. So, you get a nasal swab and a throat swab, and you wait usually 24 hours and you receive your results. That is highly accurate. However, in the early stages of the pandemic, it wasn't as common, or it didn't yet exist yet. And so when patients were presenting to hospital with respiratory symptoms, it was clear that what they had wasn't just regular pneumonia, and there was something different coming out. So there was a lot of chest x-rays being performed, and they are a fast and inexpensive way that can potentially be used for differential diagnosis. In terms of some recent studies that have investigated how accurate this is, so chest imaging for diagnosing COVID-19, some results found that it's approximately 80% accurate. So it correctly diagnosed COVID-19 in 80% of people who had COVID-19. However, it also incorrectly identified COVID-19 in close to 30% of people who did not have COVID-19. So that's, I guess, reasonable sensitivity. However, it's not a perfect measure. So that's a little bit of a caveat behind whether we should use chest x-rays or not. But there was a little bit of motivation behind using that, especially in the early days, because it's something that's fast, inexpensive, and quite routine for people who present with respiratory symptoms. Now, as the prevalence of machine learning is increasing in medical imaging, I think there's a strong need for understanding how they work and improving the trust and assurance of these models. So as an example, in 2017, there was an article published in the IEEE Spectrum, so that's the Institute of Electrical and Electronic Engineers, looking at AI versus doctors. And what we can see here is on the left-hand side, we have examples of where AI was beating doctors, and on the right, we have examples where doctors win. And in the y-axis, we can see we have increasing in time. So basically more towards the present, we can see more and more examples of where AI-based methods are claiming to beat doctors. So for example, in autism, detecting heart attacks and strokes, and most recently in this graphic, pneumonia detection. So I'm going to go into more details around this particular example. But before I do, I want to just give a bit of an overview behind some of the challenges of deep learning methods. So artificial intelligence is a broad term that encapsulates any kind of computer-based method that has some kind of signs of intelligence or can fool humans into believing so. But a big component of that these days is something called deep learning. So deep learning is effectively this massive black box where you feed in some input. You have this massive neural network with millions of parameters that then works together to produce some output. And the black box nature of deep learning is actually a major limitation that's prevented the widespread deployment of these types of deep learning models in healthcare. Why? Because we don't have any kind of reasoning or evidence behind how we generate that output. And also we have no indication of how sure the model is. Currently, we can never get 100% accuracy in deployment of algorithms. So we can't simply trust these algorithms. So as an example, you might feed in an image here of a chest x-ray. We have this black box deep learning model, and it'll produce some kind of classification. So in this case, it's predicting COVID-19. But to be clinically useful, we really want to know why. So we need some kind of evidence behind this and some explainability behind how that classification was obtained. So in terms of explainable AI, so typically shortened to XAI, or interpretable machine learning, what that means is techniques and methods that make a model's decisions understandable by people. So on the bottom here, we have two examples of what I mean by allowing people to understand a neural network model. So here we have something called feature visualization, and that looks at understanding what features a neural network is looking for. So here we can see some kind of pattern or texture. And then on the right, we have what's perhaps more easily understood, which is attribution. So given an input example, so here we have an image of a dog and a cat, we can interrogate this and figure out what parts of the image contributed to an output prediction. So in red, these are the parts of the image that contributed to a prediction of a dog. And on the right, this green region contributed to a prediction of a cat. Also, for interpretability and explainability, we need to understand beyond just the model alone, because in a deep learning especially, the model is very much linked to the data it was trained on. And so to be able to interpret a model, we actually need an understanding of the data itself. So I'm going to be going into this presentation some examples of visualizing the features, so understanding what a neural network is looking for, as well as where in the image the model is looking at. So going back to inspecting deep learning models, that particular example of pneumonia beating doctors, that was a model released by Stanford researchers called ChexNet. And it's a convolutional neural network that performs diagnosis from chest x-rays. And at the time it was released, it was claimed that AI beats human radiologists at diagnosing pneumonia. Now, if we were to inspect the model to see what it used as evidence for classification, we'd want it to be looking at parts within the lungs, and specifically parts that correspond or suggest disease. So in pneumonia, what we're looking for is these patchy white parts of the lungs, which might indicate the presence of an active infection. Now, when we actually look at that ChexNet model, so this is some research by Zek et al., released in 2018. What they found on analysis of that model was that it's not just looking at the lungs and making its predictions. We can see here, for example, it's looking at parts outside of the lungs and specifically the laterality markers. In addition, in this study, they found things like detecting the portable label and the edges of the images. So obviously, these shouldn't contribute to a positive classification of pneumonia. And it's identifying these spurious correlations that are present in the data. Looking into a couple of more examples from their paper, here we have the disease cardiomegaly, which is an enlarged heart. And here we have a color bar where these blue and purple regions contribute to a negative prediction, whereas these more yellow and green regions correspond to a positive prediction of the disease. So what we can see here is it's nice because it's looking at the lungs and it's actually correctly predicted the correct class. But if you look at the positive weights, we also have positive weights here in the top right corner where we have this label called portable. And also some positive values on the laterality label. Having a portable label shouldn't contribute to the disease. However, this model has identified that if a patient is too sick to go on a normal scanner, and that means they're on a portable scanner, they're much more likely to have an issue with their heart. So I guess this makes sense. Like if they're unwell, they're more likely to actually present with the disease, but that shouldn't actually contribute to that classification. Here again, we can see additional examples. So in this case, we can see that there's a negative contribution here. And that's because we have no portable label there. So it's down-weighted that region of the image. And so we can see again that the convolutional neural network is not just using the heart to make its decisions. Other examples include these weird high weightings on the edges of images. And it's been suggested that perhaps that's detecting the scanner that was used. So in different hospitals, there might be some kind of invisible information to our eyes, at least, that the neural network is using to make its prediction. So it's learning that association to the scanner that it was taken on and not the actual disease. And so this can result in missing the findings because of looking at the wrong information. So there's some really great work by researcher Oakton Rayner. They have, interestingly, they are both dually qualified. So they are a radiologist as well as a machine learning researcher. And what they found is that the way that this data set was collected to create this TextNet model is that the labels were generated from looking at only the text-based descriptions, and the annotators themselves never actually looked at the images. So even though the model that's being used, so the machine learning model is looking at the images, the annotators or these expert radiologists never actually looked at the images. And so that resulted in a number of flaws in this data set. And it means that effectively the labels are incorrect. And so Oakton Rayner says that I believe the chest X-ray 14 data set as it exists now is not fit for training medical AI systems to do diagnostic work. And that's due to problems with like how accurate the labels are, what the labels actually mean medically, as well as how useful the labels are for image analysis. So now looking into the actual interpretability techniques that we can use for studying neural network models. The one I showed on the previous slides, which had the grid, is a technique called class activation mapping, so CAM. And so what we do to acquire that is we make a small alteration to a neural network where instead of having a fully connected layer at the end, we replace it with this global average pooling. And what that means is we maintain the spatial information that we acquire from these convolutional maps, and then we basically sum together these kind of vectorized images to produce the output classification. What we can see from that is that because it maintains the spatial information, we can then look at these weights and then weight the maps themselves, sum them together, and it produces this class activation map. And in this case, it's looking at an Australian Terrier, and it is nicely highlighting that the input regions which correspond to the dog are being highlighted in this class activation map. One of the problems with CAM or class activation maps is that you have to make that alteration to a neural network and then retrain it for a little bit. Instead, a much more popular method these days is a method called GradCAM, where you don't have to actually alter the network, and it performs something similar. So it projects the final convolutional layer gradients back onto the inputs as a heat map. Again, nicely, we can see that if we look at different classes, so cat, it highlights the cat in the image, and dog, it highlights that as well. And so this is probably one of the most popular methods that I've seen in literature for investigating how neural networks come to their output predictions. So those methods are two common methods for for attribution. So which parts of the input corresponded to the output. Another perhaps less common method is something called feature visualization. And that is looking at the actual neural network itself and what the neural network is looking for. So most people have probably heard of something called Deep Dream. It was released by Google a couple of years back, and it was kind of this fun thing where you get a neural network to hallucinate different things in images. So in this case, it's kind of looking at dogs and kind of snout-like features, potentially because it was trained on something like ImageNet, which has a very high proportion of animals in the data set. But turns out you can apply this technique on different parts of a neural network. So you can go lower down at a channel level to get these kinds of visualizations, and all the way down onto a neuron level and see these kinds of features. You can also look at the logits level, which is before a classification on the output. And you can also look at the class probabilities as well. You can combine these techniques into something called semantic dictionaries, where you can look at parts of the image, so here the nose, and see the contribution of those feature visualizations, so how much each feature was activated. So these are actually produced by reverse optimizing an image. So rather than trying to maximize your prediction to match the ground truth labels, you basically do the reverse. So you generate an image to maximize a neuron or channel or layer. And so we can get this distribution here. So this is kind of nice because we can see that this nose kind of corresponds to nose-like features in this kind of weighting. Now, going back to COVID, there was a nice open source model that was released called COVIDNet. and it was trained on a data set called COVID-X. The details of that data set are here. So it had 3 classes: normal, pneumonia, and COVID-19, and the distribution of the training data and testing data is as follows. What you can perhaps see from this is we have a little bit of a problem that one of our classes, COVID-19, is actually incredibly rare. There's only 200 examples. So if you look at the training data, that forms just 1.5% of the whole data set. So that perhaps will give you a little bit of insight into what we expect from neural networks trained on predicting these 3 classes. Also, we have, I guess, very few testing examples, so the results may not be generalizable beyond this data set. So looking at that neural network model called COVIDNet, so that was open source, it looks like this. They basically got really good accuracy, and they also used a technique for interpreting what that neural network model has looked at for producing its predictions called GS-Inquire. And basically they concluded that because it's looking within the lungs, it must be doing a good job. So here we can see that if we look at accuracy metrics alone, we can see actually really good performance. I think this is on the order of 99% accurate, with very few misclassifications. However, that doesn't give the full picture of how this model is working. And if you apply GradCam, so the heat maps applied onto the image of what was used for the prediction, we have some examples where, again, similar to that other data set for chest x-rays, it's using these spurious correlations, so a portable label and a laterality marker, and using that to predict that this is a patient that has, for example, pneumonia or COVID-19. Again, there's other kinds of incorrect associations that the model has learnt. So for example, here we can see text labels are being used for prediction, and also often parts completely outside of the lungs are being used for the prediction, which perhaps doesn't really make sense for predicting diseases of the lungs. So we can apply some of those techniques which I described before, so combining the attribution methods and the feature visualization methods in a semantic dictionary. And so we can mouse over different parts of the image to see what features were activated as well as ones that were negatively activated. And here also we can see this GradCam, so which parts of the image are being activated. So using this, I'm going to go through a couple of examples. So first looking at correct classifications for the 3 classes and what features were used, and then looking at some of the incorrect ones. So here we have pneumonia. So this is an example where it was correctly diagnosed, and it is actually looking at the lungs. And interestingly, what we can see is that these features correspond to kind of maybe kind of rib-like features, maybe here and here. And perhaps some kind of, it's hard to describe exactly what these mean, but we can see where they activate as well. So here it's kind of the lung region and the upper shoulders, and same with this one here. Then here we have another example where it was diagnosed correctly. However, it's perhaps using the wrong information. So we can see it's highlighting the regions outside of the lungs. And using, I guess, quite unusual features, which seems to correspond to these laterality markers and kind of the central part of the image. Then for COVID-19, here we can see an example where it was correctly diagnosed, and it's looking at the lungs, which is quite nice. And we can see that the activations are also within the lungs. Here we have an example where it's correct in terms of the diagnosis. However, it's used the wrong information. So we can see that here, that it's very heavily weighting these text labels. These features are a little bit less interpretable. So it's kind of hard to imagine that this is representing text, but apparently it is. Then for normal scans, typically what was being highlighted, it was regions outside of the lungs. And so interestingly, with this data set, it turns out that many of the normal cases were actually taken from a data set, so of children. So 0 to 5 years old, the curators of this data set found some examples of healthy patients. But interestingly, 0 to 5-year-olds, what are they going to appear as? So perhaps they're going to be thinner, and that comes up in these images here. And then similarly again, so while this was correctly diagnosed, it's using the wrong information again. Now, here we have a couple of examples where it has performed an incorrect diagnosis or classification. And what we can see here, it might be slightly harder to see, but there is a medical device here in this patient. So I'm not sure if there's a pacemaker or something like that, but that's what it's highlighting on. And so it's using that as evidence for making its prediction. Now, if we look at the features that the model is using for making the classifications, what we can see is this is the channels, and this is how frequently it's observed, so that its magnitude. And so this is the most common feature across all of the chest x-rays, and this one here. If I were to describe them, I'd say this one to me definitely looks like ribs. And if you actually look at the distribution of these across the different classes, turns out it doesn't actually vary very much. So basically what it's telling us is that the neural network models are always looking at the same features. So basically looking at, is it lungs? Is it, perhaps that kind of white fuzzy appearance? But that's not really that useful when we want to analyze the properties of a disease. So what we then did is modified the standard semantic dictionaries to look at class-specific features. So in this example, we have pneumonia, and what we can see, interestingly here, it actually came up with these kind of more patchy features in terms of what it was using for its prediction. And then also interestingly for COVID-19, if we look at just the features that are specific to that disease, it's weighting more these kind of, I kind of describe it as noise. It's kind of hard to say what this actually is. And perhaps it also could be an artifact of the data set itself. But the nice thing is you can see how the neural network changes what it's looking at depending on the class label that you specify. So in just the raw activations, we can see that it is highlighting those two very common examples I showed before, kind of equally. But if we look at the different diseases, we can see that it's looking at different types of features to make the classification. So in all these cases, this is the same image. And so interestingly, it's looking for different features for the different class labels. And so that was combined together into this kind of nice visual user interface tool. So you can mouse over the regions. So the regions it thinks is important are in red, and we can see what those features actually look like. And this perhaps gives us a bit of a better understanding of what neural networks are actually doing in terms of diagnosing or classifying chest X-ray images. Now, as I briefly mentioned just before, turns out there are actually quite significant problems with the data set that this COVIDNet model was trained on. So it turns out COVIDX was actually composed of various other data sets. So we have these ones here, so that's all fine. However, once you go to here, we can see a couple of problems. So one, this data set here for healthy normal patients was collected from children aged 1 to 5. So there's going to be this kind of bias present in the data because in all of the other classes, for example, COVID, it'll never be a young patient. Then we can see these arrows, which are going in multiple directions. Turns out these data sets actually appear multiple times because different sources actually included it twice. So Cohen includes SIRM, as does Chowdhury. And so this data set, what that means is we're going to have data duplication. And also, it is going to result in data being in both the training and test set, which is a big no-no in machine learning research. So that is, contaminating the learning process, and effectively it can cheat and memorize those examples. It means that the results will also be wrong. And like I said before, those biases that are present, the model will learn to distinguish adults versus children, but not actually detect the disease that it should be detecting. So this is something that's been coined Frankenstein data by an actually quite a good paper on COVID-19 detection. So common pitfalls and recommendations for using machine learning to detect and prognosticate for COVID-19. So that was published in March last year. So one thing we looked at, so taking that model that we had, the COVIDNet model, but looking at more data. So perhaps, we can kind of overcome that bias in this small portion of the data set that had the normal young patients. So here we can see that we have a lot more examples. So in some cases less, but we have significantly more COVID-19 examples. And retraining that model, we get this nice diagonal. So it's actually performing the classification correctly. Looking at the distribution, we have now a lot more COVID-19, so hopefully we can get a better diagnosis or detection of COVID-19. But if we use alternative interpretability techniques, so this is perhaps the most simplest thing that you can do in terms of understanding your data set. So what this is here is looking at the raw pixels of every image. So let's say it's for example, a 100 by 100 image. So that is 10,000 values, perhaps RGB or not. You can then project that down into 2 dimensions. And that means that the spacing here between points is how similar they are in pixel space. And here we've got these colors for the different diseases. So normal in blue, pneumonia in white, and COVID-19 in red. What's unusual here is that we can see some kind of clustering happening. So looking at the raw pixels, we don't expect to be able to diagnose diseases. If we could, it suggests that there's something wrong with the data collection process. And so what I can show you now is that as a animation. So hopefully you can see this. So we can now mouse over these examples and see what they look like. So we can see what the correct label was and the source of that data. So we can see that there perhaps are some differences in these images. So here especially what we can see is that we've got this really strong black outside the image. In these images to the left, here we can see similar, but maybe those particular scans have maybe larger hearts or something. Here we have another cluster. Those images look a bit more gray. So what we can see here is clearly there's some biases in the way these this data was collected. So that's the finding labels. We can also look at the source of the data. So it turns out this particular data set was collected from, I think, 5 separate data sets and combined together. So here we can see that those finding labels and the source of the data are actually quite correlated. And this is a bit of a problem. It indicates that our machine learning model will have to somehow overcome these biases in the way that the data set was curated. So here's an example of that there. So going from looking at the correct labels and looking at where that data was collected. So COVID was collected from 4 different sources. The normal class was collected from 1 source and pneumonia from another source. And so this, just looking at a very, very simple distribution of the data set and coloring it in by where it came from, we can see already that there's quite big issues in the data. So to summarize some of the problems in COVID diagnosis from chest x-rays, what we can see is that what we typically use for comparing models, so accuracy or perhaps even sensitivity curves, so precision recall, that alone is not a good metric for understanding performance, particularly in generalizing to new data. And by using these kinds of interpretability or explainability techniques, it helps us to visualize and understand both the model as well as the data that was used to train the model. I think a big conclusion from the chest X-ray machine learning models is that we really need well-curated data sets. So, garbage in, garbage out for a machine learning model. And so, first of all, if you have a lack of data, so in the example where we only had 200 examples of COVID to learn from, obviously to be able to perform that, the model is going to have to do some kind of memorization and learn these incorrect correlations that are present in the data and result in a heavily overfit model. In addition, how the data sets were put together is also really important to understand. So as we saw in the COVID-X data set, we had that contamination of the data because we had training and testing examples, so the same image appearing in both. And also, probably more strikingly, we had children in the normal class where we didn't have them elsewhere. And so that introduces biases into the data, which then our model is going to learn and then produce incorrect correlations, which then result in incorrect classifications. And I think even more importantly is we can't have machine learning researchers perform this modeling alone. We really need medical experts to guide the full process of a task, all the way from data collection to avoid those issues with incorrect labels, as well as contamination of different data sets appearing multiple times, incorrect labeling, as well as defining the task itself. So interestingly, we don't actually diagnose COVID from chest X-rays. And in fact, it's not actually able to perform a very effective differential diagnosis. For example, many people don't actually have any respiratory problems, but they still might have COVID. So we have to really think of the clinical use of these types of models as well. And in some of the analysis I've showed, what we can see is that the data has been labeled after intervention. So perhaps I didn't have the exact examples, but interestingly, in a lot of the COVID and pneumonia, the patients have already received a chest tube to help prevent or alleviate some of their symptoms. And that actually comes up in the modeling. So rather than detecting the disease, it's detecting that this patient has been already treated for a disease. And that treatment is not a sign of the disease itself. And so explainability and interpretability helps us in terms of understanding how these models work, understanding the data distributions as well. I also want to raise the importance of not just explainability, because alone, I don't think that is the solution. What we really need is something called reviewability, which helps in exposing the decision-making process. So if we have the full process able to be reviewed and actually detail it to others. So for example, if we know what decisions were made by the engineers in developing the system, if we know the data that was used to train and test the system, how we actually went about training and testing, and then also the fairness, the effects and lawfulness of the automated decisions in practice. So that's also really, really important. So that allows us to then build up other people's research as well as have a full understanding of the full process beyond just looking at, for example, accuracy metrics or these featured visualizations. And so in conclusion, I'd also like to acknowledge my collaborators again. And so thank you very much for listening to my talk, and I'm happy to take on any questions.
Any questions from the audience?
Maybe Sally.
I've got a quick question. Thanks, Alina. That was fantastic. I'm just wondering about whether, and I can't remember if we talked about this, but this problem that the model's learning things outside the kind of field where it should be looking, whether you can help the models in the training process by doing some segmentation prior to it. So for example, there's some pretty good segmentation algorithms out there to pick out the lungs. Yep. And then the model only, you block out the rest of the image.
Yeah, definitely. So even interestingly, in the COVIDNet paper itself, they actually did do cropping of the outside. So it helped to some extent, but even in terms of looking at the explainability, still there's there's parts that still appear. So especially with the medical devices, I think that's something that we can't easily get rid of. And that's, yeah, in terms of looking at the lungs, we can direct a model to the lungs. And especially some models, they look at patches instead of the whole image and then focus only on the lungs, where you can actually guide it to only look at the lungs. So definitely one of the potential solutions for getting rid of that spurious information on the edges.
Right, thanks.
Thanks, Ellie.
I have a few questions. Thanks for a very nice talk. So three separate questions. Try to be quick. So the first one is if we just think of medical imaging in its own right, I mean, this is already in a form, some sort of feature creation where, you know, we as humans, we weren't naturally born to see MRIs. Right, so the radiologists have trained with a lot of knowledge to understand what an MRI looks like, etc. So are there directions of kind of taking inner features, inner layers, or other representations, and then creating a different form of representation for the radiologist that still has all the subject matter knowledge? You know, so not kind of trying to take the radiologist completely out of the loop, but rather enhancing the image.
Yeah, I don't know specifically research that does that, and I think it's quite hard to do. But I think that actually makes sense, because when we're generating these, you know, interpretability maps, you know, even, I mean, to non-experts in medical imaging, they don't mean very much. I can kind of see it looks like, you know, maybe ribs. So it really would help to have that direction from experts on what they actually look at and what's useful. So I don't know of specific research that does that, but I think it's, you know, worth looking into.
Thanks. I've got two more questions, but maybe somebody else wants to ask something, and then I'll ask again.
I've got a question. Thanks, Alina. That was super interesting. It's not really my area of expertise, but super interesting. So I guess in real life as clinicians, before we even look at the x-rays, we'll have other information that helps us, you know, priors, if you want to call them, like age and sex and the symptoms and various things. Do you ever incorporate those into your models that, you know, that just look at patterns?
Yep, definitely. So I 100% agree with that. So, you know, looking at an image in isolation is actually incredibly challenging. I think even for radiologists, they usually have some kind of text, you know, to kind of guide what they should be looking at. So there is research in that space. I think it's just sometimes hard because, you know, that introduces its own biases as well, and whether we want to or not. It's you know, an interesting question, because, you know, if we tell it, okay, we're looking at this and we expect it to be this, will it only look at that? Will it miss other things? It might introduce other biases perhaps, but it is something that people are working on. It's just challenging because it requires that multi-domain expertise. So even in this space, most of what I've been looking at is images, but a lot of the other information is in different modalities like you know, text. And so you need natural language processing to understand that. And so some of my colleagues are actually looking into that. So how we can, you know, extract information from text-based reports. So basically completely unstructured data, and how we can extract information from that. And then combining different modalities is also quite challenging, but definitely something that would be useful because that's what clinicians themselves do.
Thanks, Alina.
Yeah, thanks for that.
Yeah, I think Yoni, you can ask the second question.
Okay, thanks. Yeah. So about GradCam. So if I understand, you use GradCam to find features in the original input channels that are most dominant in kind of making the prediction.
Yep.
So in a statistical point of view, one might try and kind of, if you think like of mixed models and statistics, one may try and maybe remove those features and then look at what's left, right? So say you have dominant features that discriminate over age, it's clear that age is a major factor in COVID-19. So you remove those, mask them out, and then you go further.
Yeah.
Is there any hope or work on that?
I think that's a great idea. In my past work, I've done a lot of that. So it falls in the space of perturbation interpretability methods. So basically, you'd have to, you know, corrupt that part of the image which it thinks is important. So, you can remove that information and see how it predicts without it. In fact, you can do that completely without GradCam as well and get its own kind of visualization. So if you literally just blank out each part of the image and then see how it affects the prediction, we can identify regions that are important. And so you could do similarly where you remove those parts and see the importance. The only catch is doing it wide scale. So like on a data set, so the chest X-ray 14 data set is around 60 gigabytes. And so even just processing that all with the neural network, it takes, on the order of a couple of hours. If you introduce perturbation methods, they're probably on the order of like 8 times more expensive. So something that might take a day is going to take, 8 days. Admittedly, computing resources are increasing quite significantly. So it's definitely a possibility. I think it's just from my own research, it's like you want results relatively fast. But that is in terms one of the things that we're looking at kind of in the next phase of looking at chest X-ray analysis.
Ian has raised a question. Yeah.
Yeah, hi. Thanks, Alina. Very nice talk. I just want to comment a little bit on the data side of things, if you can hear me.
Yep.
Just so, it feels like a sort of an old issue where if you want the accuracy estimates you're getting on test data to mean anything for the population you want to apply it to, then your data should have come from that population. So if you're going to be grabbing data from various sources without thinking about that very much, you're going to run into trouble. And so, but of course, these are not really designed experiments. These are repositories that you're kind of coming across and trying to make good use of, which is fair enough. So I think in image analysis, there probably have been efforts to standardize images typically. I guess that has to be done pretty carefully to try and avoid these, spurious features, let's call them. But yeah, you've done well to highlight them visually, but I guess it must be a little more difficult when the when it's not image-based, when it is all entirely non-image type of predictor variables, but still, anyway, I guess you could use similar types of methods.
Definitely. There's a whole host of features. I think the kind of GradCam and class activation maps are probably more suited for images, but there are a whole host for text-based analysis as well. So I think so Sally's actually done a lot of research in that herself with a number of postdocs and research students. But yeah, the data problem is something that's incredibly challenging because acquiring data sets is quite difficult because you've got all of the privacy and ethics around the data collection. And so what people usually do is find stuff on the internet and put it together. And so that's probably actually in this case the most important factor in the output results, but it's actually the part that a lot of researchers don't have any control over. And so in some respects you need to have like a collaborated effort towards putting these researchers together, so the people that do the data collection through to the people that do the analysis, because you really do need the full process understood.
Yeah, it's common to a lot of areas where they have different equipment or different ways of the lab, and they try to adjust them all so that they correct for that lab effect or the type of equipment. Yeah, it's a process. It takes a while, especially if there hasn't been much collaboration in the past.
Yeah.
Okay, well, good luck with that. Yeah.
Thanks.
Okay, Yoni, your last question, third question.
Oh, thanks. Yeah, sorry. Yeah, I think you might have answered it a bit with saying that GradCam is computationally much harder than just the prediction algorithm. But my question was if you can just kind of say the difference between, if you do image segmentation, kind of pixel-wise segments, which is sometimes used in medical imaging, versus doing this kind of classification and then going back and reading the pixels. How do these methods interplay? Or are there alternatives or whatever?
Yeah, I think some streams of research are looking more into symbolic AI, so like things that you can actually quantify. So like they'll do segmentation. They know this is a heart because that's what the segmentation model says. They know this part is the region that corresponds to the lungs. But yeah, I think there is some research in that space. I think a lot of people that have done this research in this work, myself included, it's because maybe it's easy. You know, you're like, okay, let's just train a neural network and see what it gets. And in fact, people have got really good results in terms of accuracy just doing that. But I think, you know, we need to look beyond just, you know, looking blindly at the image. And I think more and more people are looking into that. So, you know, focusing on, you know, segmenting the lungs first and then only looking at those parts. So that is definitely something that a lot of people are doing. It won't have as interesting, you know, problems and flaws that we see here.
Okay. Yeah, great. Thanks.
Yeah. Thank you. Any more questions? I've got one. Okay, so like in terms of like the application of this work in actual like diagnosis, what would be the role of the radiologist? Like, can they use this?
Yeah. So interestingly, for a radiologist, the interpretation of the images isn't the only task that's performed. So there's kind of like a whole suite of things that happens, but what we can see is at the moment, these models aren't really 100% ready for deployment. Perhaps, you know, in a couple of years' time, we'll have models that know when they're accurate and like maybe they're easy cases and it'll be perhaps easier to skim through. I don't know if that's going to result in, you know, missed diagnoses or not. But, you know, perhaps it can, you know, focus radiologists' efforts on the cases that, you know, are more difficult. So ones where we're not certain, ones which are more difficult to diagnose, but in some respects, it might just help them to know where to look. So saving a little bit of time, but really we need the to have the proper, you know, trust in these models. So actually have, you know, proper accountability. So understanding how the data sets were generated, understanding how they work. Before being used. And so I don't think radiologists will be replaced anytime soon.
Thanks for the excellent talk. Any more? We are running out of time. Like we can have quick one more question if there is one. No. Okay. Can we all thank Alina for her talk? Thank you so much for joining us.
Yeah, thank you very much for having me.
Yeah.
See you later.